We wanted just really to announce some very exciting news that has literally just come out today, hot off the press. Niklas is going to take you through that. So please feel free to ask us any questions afterwards. And ultimately, we'd love to help you with your products. So Niklas, over to you.
Thanks, Christian. So yes, as Christian said, we had some nice news this morning in a press release, and you can share on the screen now. So we have, as you might know, we have conducted a clinical study, and we have additional positive interim results from that study. And what we saw in this basically study is that our nanoparticles, our unique nanoparticles, might offer viable alternatives to complex formulation approaches, such as cyclodextrin-based technologies. What we did in the first study, we showed that we have a faster absorption of our nanoformed CESS particles and the formulation against Felden, the reference product marketed by Pfizer. And the second study, we compared the nanoformed Piroxicam tablet formulation against beta-cyclodextrin coupled Piroxicam Brexidol. And what we saw in the study was that we showed equal absorption performance when comparing to the Brexidol, which gives us really good confidence that we can offer a solution with much more simple formulation. So basically what we have done is used pure API nanoparticles in a very simple traditional formulation, and it performs as well as complex formulations, which is good news in terms of the fact that we can work with the simple formulation, and we will not encounter such safety and tox concerns that there are often with some more complex formulation ingredients. So this is the good news, and I think we can go over to a video now before my presentation, showing how the actual clinical study was done and what we have been doing in the clinic with our nanoparticles. Thank you.
Nanoform is a nanoparticle medicine-enabling company that works to facilitate life-changing medicines for patients around the globe. Our award-winning nanoparticle engineering technology, controlled expansion of supercritical solutions, or CESS, enables us to nanoform drug substance particles to improve bioavailability and solubility. By doing so, our technology addresses a leading cause of failure in the drug development pipeline, with a significant proportion of new drugs failing due to poor solubility. Through our unique nanoforming process, we aim to double the number of novel drugs reaching the market and touch the lives of 1 billion patients globally. We partnered with Quotient Sciences to create a nanoformed formulation of the anti-inflammatory drug Piroxicam for use in human trials. By applying our technology, we may be able to significantly improve the pharmacokinetic properties of the drug and potentially reduce both the required dose and side effects. By showing the value of our technology to enhance this popular therapeutic, we are one step closer to nanoforming the medicines of tomorrow on a large scale. Our main goal is to give hope to patients suffering from currently incurable diseases and to help more people around the world access life-changing treatments.
The journey of a nanoformed drug candidate begins as powder of the raw material, which we convert into uniformly sized nanoparticles using our CESS process. Initially, we dissolve the Piroxicam bulk powder in supercritical carbon dioxide, an environmentally friendly solvent. By carefully controlling the temperature and pressure, we achieve stable nucleation. The nanoformed API particles undergo rigorous testing to ensure that they meet GMP requirements.
The successfully nanoformed Piroxicam is carefully transported to our partners at Quotient Sciences, who will manufacture a tablet formulation of the drug at their state-of-the-art facilities in the UK. Nanoform's partnership with Quotient Sciences will culminate in the first ever human trial of a nanoformed drug candidate.
To overcome the long-standing challenge of poor efficiency in pharma, we as an industry need to pool our expertise to develop and implement innovative solutions for patients. We created our CESS technology for precisely this reason, and the partnership between Nanoform and Quotient Sciences represents a significant milestone in Nanoform's history. We look forward to continue working with industry partners to help life-changing drugs reach the patients who need them.
Good afternoon, ladies and gentlemen. It's my pleasure to give this talk, Small is Powerful, the Nanoparticle Solution to Big Bioavailability Problems. I will talk to you first about the company a little bit, give you an introduction to the topic, then I'll dive into the CESS technology, give you some case studies, and finally after the talk there's a possibility for you to ask questions. Poor bioavailability and low efficacy are the most common reasons for drug failure, as you see on the graph on the left-hand side. Additionally, we can see that 70 to 90% of new drugs are poorly soluble, which means that they fall into CESS class 2 and 4 drugs. And really our mission at Nanoform is to enhance the pharma industry output by targeting the poorly soluble drugs. And also lately, we're bringing a solution to the biologics field for drug delivery applications in that area. I will talk about that later on in this talk as well. If we look at the importance of particle size in tackling the bioavailability issue and the poor solubility issue, is presented on this slide, and we can see that smaller particles have a larger surface area, and a large surface area of particles enables better bioavailability of a drug. So if you look at the graph on the left-hand side, we can see that if we decrease the size from 10 microns to a particle size down to 100 nanometers, we get a 30-fold increase in the surface area of a drug. A further reduction to 50 nanometers gives an increase of 1,000 times in the surface area, which is of course very important for improved bioavailability, and it will imply better absorption in the body. Our CESS technology really addresses this issue because we are nanoforming our particles and increasing the surface area to improve the bioavailability of the drugs. If we look at the nanocrystals in general, some of the advantages are, as I just mentioned, the increase in surface area. We also increase the saturation solubility, we decrease the diffusional distance, and increase the concentration gradient, which all are factors that will facilitate increased dissolution velocity or dissolution rate. So this is important to understand. Something about Nanoform, we're currently about 70 employees and growing. We're very international and we're science-based. We have more than 25 PhDs from different fields, and this is really the strength of our company. We have a great team. Here are all the faces of our team members. We strive for innovation, quality, partnerships, and transparency, and we're really happy to have a growing and motivated team of people. And this presentation partially depicts some of the work that we have done over the past few years. So let's dive into CESS technology. CESS, controlled expansion of supercritical fluids. Basically, it's a drug substance crystallization process using supercritical carbon dioxide. So on the left-hand side, we see the process schematic. We start with liquid CO2, which is guided into a pressure vessel where we load the API. We increase the pressure and temperature to reach a supercritical state of the CO2, which makes the API to dissolve. And through this chamber, we guide the solution towards a collection vessel with accurate control of pressure and temperature and flow rate, which allows us to initiate nucleation of particles and particle growth and collection of the particles in the collection vessel. In that phase, CO2 also expands to dry ice, which basically means that we are covering all the formed nanoparticles in dry ice, which is a further controlling step of our particles. Then the dry ice is sublimated. We remain with the pure nanoparticles. Here you see a picture of micronized particles turning into nanoformed particles. Typically, depending on the customer requirements, typically we produce particles between the range of 100 to 800 nanometers, but we have shown that with certain APIs we have gone down to 10 nanometers. This is a short video on the last step, the collection phase of our process. So here you see the dry ice forming, and within this dry ice, the nanoparticles are embedded, and when the dry ice sublimates, the nanoparticles remain and can be used further for downstream processing and formulation studies.
We work in a structured way. We basically, when we get APIs in from customers, we have a star map tool, which is basically an AI-based tool to basically predict the success of nanoforming. That's what we use first, and then we have a basically for a proof of concept studies, we do a thorough physicochemical assessment, manufacturability assessment, and a risk assessment, and then we run a proof of concept study, a systematic design basically to understand is the API nanoformable and how well it performs. Then we can enter to a proof of process stage, where we basically optimize the process and establish a design space and are ready for technology transfer to GMP if the projects advance in that manner. A few words about StarMap. Basically, we use input properties of molecules, feed them into our AI tool, and we are able to predict solubility in supercritical CO2 and the propensity to crystallize. And these are, of course, key to understand how we should run our process. Then let's dive into some case studies here. So we have a reproducible process. We have shown this over and over again in our with our APIs and customer APIs. Here you can see Piroxicam. We can show high batch consistency and high long-term stability, and we have demonstrated that if, for instance, in this case, there was no change in crystallinity, particle size, and chemical purity of these 200 nanometer or sub 200 nanometer particles that were produced. Here we have a couple of videos that you can see the nanoformed particles. The handleability is of course important, and it is interesting. We're forming these highly or very small particles, and one would think that the handling of these can be very difficult. But as you can see here, the nanoparticles actually form loose aggregates, which are then easily, basically, you can handle them easily. But then we can show when we do formulation studies, for instance, that they are redispersible in suspensions. We can easily redisperse them in powder blends and so on, and make tablets out of these. I'll show you a few examples of these here in the next slides. So here are some examples of suspensions. Basically, Piroxicam again, the nanosuspensions have shown to be very stable compared to reference suspensions in this case. Where we have micronized Piroxicam, they are easily redispersible. If we look at the particles, the primary particles in the nanoform suspensions, here we can see on the left-hand side the two images are the nanoparticles extracted from the suspension. So we can see that the particles remain in suspension with the same morphology and particle size, and the nanoform bulk dry powder is shown on the right-hand side, so we can compare these images. Then to a very interesting study that we have performed on rats. It's a PK study with nanoform Piroxicam, 200 nanometers, and a very tight span. We compared it to micronized API with a D50 of 2.1 microns and with a larger span. And what you can see is that the nanoform Piroxicam had improved pharmacokinetic properties compared to the reference API. And you can see that the Tmax is faster at the time which maximum concentrations is observed, the Cmax is higher, and the AUC is larger. And if you compare the micronized reference to the nanoform, the difference is very significant. Interestingly, we have we also studied a lower dose of the nanoformed particles, so a 5 milligram dose as opposed to the 20 milligram dose per kilo per orally. And you can see that we come basically halfway the micronized material with the 5 milligram dose as compared to the 20 milligram dose, which gives us really nice results in terms of having the potential to reduce the dose of treatments with our nanoformed material going forward with our customers. A few words about tableting studies very quickly. When we did standard tableting compression studies, there was no difference in the compactability between placebo, nanoformed, and reference material of micron-sized Piroxicam tablets. And this is good news in terms of handleability again, or downstream processability. So basically what it means, nanoform material can be formulated with traditional methods, and there is no need of complex techniques for our particles. And looking at this integration, the specifications were fulfilled as compared to pharmacopoeial specifications for IR tablets, very fast disintegration, and a in vitro dissolution study compared to a reference commercial compounds Felden and Brexidol, and a generic product. Our nanoform tablets have faster Piroxicam release compared to these products, as you can see in the graph on the right-hand side. We saw the video on the Quotient Sciences collaboration, where we are dosing nanoform material at the clinic, and this is very exciting times for us, and we're expecting results by the end of basically before the end of Q2 2021. So this is very exciting times for us. Let's then jump into another case study here quickly. I showed you an example of a crystalline material. This is just to show that we can also make stable amorphous nanoparticles, in this case sub 50 nanometer particles, and these particles have been stable for a long period of time, and the results are reproducible. And you can see the nanoparticles, how they behave in vitro dissolution compared to the bulk material. The difference is huge, and basically a non-existent dissolution for the bulk material, whereas the nanoparticles dissolution rate is very much different. So here is just briefly 6-month stability data, an example where we can see that the particle size is consistent, and we can show stability. Here we have XRPD data below the SEM pictures, and we can see the amorphous halo of the particles that we have been studying, and the dissolution remains the same also throughout this stability testing period. So it's very exciting that we can also, with this technology, make these confined small amorphous nanoparticles that are in the best cases very stable. This is an exciting study, a recent study. I will only show you this slide, but we compared our CESS technology with other approaches, namely the standard approaches that are used for poorly soluble drugs. Spray-dried amorphous dispersions, hot melt extrusion, salt formation, co-crystal formation, and milling and micronization of the API. Again, in this case, we looked at Piroxicam, as you can see, this is a suboptimal dissolution method, but it needed to be developed in this way that we can get all the graphs in the same picture. But you can see that our nanoparticles outperform the other techniques by far. So this is exciting news for us, and we can talk about this later on with you in separate meetings. So very briefly about development of manufacturing capabilities. The GMP plant is basically in Helsinki, and you can see some of the pictures of the facility here. We have non-GMP lines or R&D lines, eight of them, to support customer projects, and then we have our first GMP line, which has produced the clinical material for the study that I just mentioned, and that was shown in the video before. We of course want to build in quality into our products, so we use a quality by design approach in Nanoform, and you can see it here. I won't spend too much time on this today. Finally, some exciting news from the biological side. So what I just talked about before is the small molecule, basically nanoforming capability, the CESS process. But we have 2 weeks ago announced our maturity has increased in our another platform that we have been developing over the years, and this is a proprietary nanoparticle formation technology to deliver biological nanoparticles, and we have shown that we can go down as small as 50 nanometers. We have filed a patent application with the US Patent Office, and we have 2 R&D or non-GMP manufacturing lines at the moment on the biologics side. And of course, what we believe that we can bring to the table, we believe we have a game changer in biologics. We can start addressing the improvement of delivery routes from IV to subcutaneous and oral routes. We believe we can make a difference in the uptake of biologicals and biodistribution of biologicals. Increase the drug lowering capacity in formulations, enable new types of drug combinations, and tailor release profiles, and also maybe bring something new to the basically the cold chain or cold chain needs of biologicals. Maybe lighter infrastructures for the logistics with our particles that we can produce. So basically, the process is a gentle bottom-up process, and it's scalable. We have worked so far with different types of proteins ranging from 6 kilodalton to 60 kilodalton, and we have shown that the biological activity is maintained in our systems that we have looked at, and we're now really looking at partnerships with pharma and biotech to start working with this novel technology. Here are some images. Lysozyme, it's a typical model compound, but you can see we have nanoformed lysozyme into, in this case, about 150 nanometer particles. These remain their biological activity. We have two different types of process lines, and we can that allows us to tailor the particles as well. Here you can see a few examples of slightly different particles. Finally, a summary, and I apologize for the technical problems, but I hope you have gotten a picture of our technologies, both on the small molecule side and the biologics side, and we bring pharmacokinetic improvements into the game with our technologies. We can address many additional drug delivery applications apart from the ones that were shown here, and of course all the commercial opportunities that come with this technology, from enabling new drugs to progress into the clinic, life cycle extensions, additional patent protection, and so on, will make a difference then for our partners. The benefits of CESS, I hope you got them in this talk already, so I won't be repeating these. Finally, I just want to say thank you, and we have a strong team that you can contact. Sergi, Christian, Gonzalo, and Eric are available, and myself, of course, to answer questions and talk more about collaborations in between you and us.